Assalamualaikum and good morning everyone. So today we continue with gene expression. Okay, so first things first, let's study about Beadle and Tatum's experiment. So apparently this is the late Mr. Beadle. Okay, this guy here, George Wells Beadle, and together with Edward Lowry Tatum. Okay, and this if you notice, okay, on the screen there are this red pinkish colorish fungus. Okay, this is the Neurospora crassa, which can be found on your expired bread, or they are commonly known as the bread molds. Okay, gene expression. Gene expression is a process by which information from a gene is used to synthesize of a functional gene product. So meaning to say, gene expression is when your DNA, your gene on your DNA, okay, is being used to synthesize a product. Okay, it's normally a protein product, such as enzymes. Your enzymes are proteins. So Beadle and Tatum experiment concluded that one gene, okay, controls production of one enzyme. So this hypothesis is known as one gene, one enzyme hypothesis. But later on, it was amended to one gene, one polypeptide hypothesis, because synthesis of two polypeptides in a protein, example your hemoglobin, okay, we have the alpha chain and the beta chain, or the alpha polypeptides and the beta polypeptides, okay, can be controlled by two genes. So apparently there are two genes that control separately the synthesis of the alpha polypeptide and the beta polypeptide. Now, in Beadle and Tatum experiment, they observed that the mutated spore of the Neurospora crassa can only grow in a minimal medium with arginine, okay, because one gene has mutated. So the mutated gene cannot direct the synthesis of enzyme that is related to the synthesis of amino acid arginine. Can you get what I mean? Meaning to say, okay, the mutated neurospore, okay, Neurospora, okay, it by right it can grow on minimal medium. Okay, I explain further on that later. But what happens? This mutated, they cannot grow on minimal medium. They can grow on minimal medium, but that minimal medium must have arginine, and arginine, okay, is an amino acid which is needed for growth. So what happened? That one particular gene in the mutated spore, okay, cannot synthesize, okay, or cannot code the synthesis of enzyme. Right? Why it cannot code the synthesis of a particular enzyme? Because it cannot code the synthesis of amino acid arginine. Now, the principle used in their experiment: the spores of mutated wild-type fungus Neurospora crassa cannot grow in minimal medium, okay, because the lack ability to synthesize its own amino acid. So if it cannot synthesize the arginine, okay, the mutated type of Neurospora crassa. So what was done by Beadle and Tatum? So first, Beadle and Tatum, they irradiated Neurospora crassa spore. Okay, the purpose is to induce mutation, or to cause the wild type Neurospora crassa to become mutant. Next, the mutated Neurospora crassa are cultured on a complete growth medium containing all amino acids, vitamins, and other nutrients. Wild-type Neurospora crassa can grow on a minimal medium. Alright, what is minimal medium? It's a simple solution of inorganic salts, glucose, and biotin incorporated into agar. Right, agar is agar-agar lah. Okay. Alright, so from this diagram, as you can see, okay, the Neurospora crassa that is irradiated, okay, with UV light or X-ray, they can grow. okay, on minimal medium. Beadle and Tatum identified nutritional requirements in mutant strains by testing for growth on minimal media, supplemented with individual vitamins or amino acids. Okay. So what happened? Okay, Beadle and Tatum, they want to identify, okay, on minimal medium which that has certain a nutrient, okay, will induce growth, will cause growth in the mutant neurospora. So they transfer some mutant fungal cells or some of the mutant neurospora crassa to each of these 3 tubes. Apparently in the original experiment there are 7 tubes. So to simplify learning, okay, we just focus on these 3 tubes. Okay, so tube with minimal medium plus vitamins, okay, as you can see from here, and then tube with minimal medium, okay, with amino acids, and tube with only minimal medium without any other supplements. Okay, so what do you observe here? The mutant fungus will grow on the minimal medium supplemented with amino acids. Okay, so next, to determine which amino acid the mutant fungus had lost the ability to synthesize, Beadle and Tatum transferred the mutant fungus, okay, to tubes containing minimal medium with only one type of amino acid only. Okay, so there were 20, if I'm mistaken, because we have 20 types of amino acids, right? So what happened? Every test tube, okay, they put in the minimal medium with only one type of amino acids. Okay, and then they insert the mutated neurospora. Earlier, they want to know this mutated neurospora can grow on minimal medium with vitamins or with amino acids. So they concluded it can grow on minimal medium with amino acids. It can't grow with it can't grow in minimal medium with vitamins. Now since it can grow, the mutated neurospora can grow on minimal medium with amino acid. They want to identify which type of amino acids. By right, this mutated neurospora cannot grow on minimal medium, but it able to, provided there is certain amino acid being added in. So this is the test tube with amino acid, minimal medium with arginine, okay, and then test tube with minimal medium and tryptophan, lysine, leucine, okay, glutamic acid. There are many others amino acids, right? So what is the outcome? What was the outcome? So Beadle and Tatum found out only the medium containing the amino acid arginine supported growth, and this indicates that the mutation affected some part of the arginine's biosynthetic pathway. Okay, or I can simply say the mutated spore can only grow in a minimal medium with arginine because one gene, okay, has mutated. So that mutated gene cannot direct the synthesis of enzyme that is related to synthesis of amino acid arginine. You get what I mean? Let me rephrase that. One gene in the mutated neurospora, okay, cannot direct the synthesis of an enzyme, okay, and the synthesis of this enzyme is needed, okay, to synthesize amino acid arginine, which will support growth. So Beadle and Tatum concluded that each gene controls the production of a single enzyme. Okay, that's It's about Beadle and Tatum experiment. So test your knowledge. There are two questions here. The experiments with nutritional mutants in Neurospora by Beadle and Tatum provided evidence that, so we have A, B, C, and D options here. What do you think the answer is? Bread mold can be grown in a lab on minimal media. No. Okay, because this experiment involved mutant. X-rays can damage DNA. Can. Yes, it does, but this is not the main purpose of this experiment. Cells need enzymes. Yes, again, this is not the purpose of this experiment. Okay. Genes specify enzymes. Yes. Okay. Genes coded synthesis of specific enzymes, and these specific enzymes will coded the synthesis of polypeptide or specific certain protein. So the answer is D. Okay, gene specify enzymes. Okay, describe Beadle and Tatum's experiments with Neurospora. So those whatever I've explained earlier is not the full version of the experiment, but it's sufficient for your level. Okay. You want the full version of the experiment, you may refer to the Oxford and Pelangi textbook, which I believe is a bit complicated. Okay, and there are many unwanted informations there. Okay, unneeded, you are not you don't need it for your level. Okay. Okay, so now 5 marks for this question. I give you 10 minutes. You read back the module, okay, and then try to answer this question. Okay, pause this video. After 10 minutes, then you come back here. Alright, see you in 10 minutes time. Okay, 10 minutes have passed on. Wow, that was quick. Okay, so let's proceed on the answer for this question. 5 marks. Okay, so point number 1. Okay, Beadle and Tatum exposed Neurospora exposed to X-rays or UV radiation. Okay, Neurospora crassa must be underlined. And point number 2, why do they expose the Neurospora to X-ray? To induce mutant strain or to induce mutation in the Neurospora. And then P3, and then identified strains that carried a mutation, preventing the fungus from producing an enzyme essential for growth. Point 4, their work or Beadle and Tatum experiment revealed that each mutant strain had a mutation in only one gene, and that each gene affected only one enzyme. Okay, that's it. Now let's continue with genetic code. What is genetic code? It is an inheritable information and characteristics of an organism that's stored in DNA, held in the order bases along section of the DNA. Okay, so genetic code, they are being held in order, okay, of bases along section of the DNA, and this section of DNA is known as cistrons and contain information to synthesize particular polypeptide. Now, class, apparently on a gene, on a DNA, okay, there are bases that is readable for synthesis of polypeptide, and there are bases which is it does not code synthesis of any product at all. Okay, so the ones that code the synthesis of able to code the synthesis of product is known as cistrons. Okay. Genetic code consists of 3 bases, okay, which is called triplet codes. What is triplet codes? A genetic information system in which sets of 3 nucleotide long words specify the amino acids for polypeptide chains. Okay, and these 3 bases of these triplet codes can be found on DNA. Now, let's take a look on the triplet codes characteristics. Sorry, not triplet codes, genetic codes. Apparently, this genetic code on DNA, they are known as triplet codes. On mRNA, they are known as codons. On tRNA, they are known as, they refer to genetic code, are known as anticodon, okay, on tRNA. So it has different names on different molecule. Okay, on DNA is triplet codes. Okay, now characteristics of this genetic code. Each triplet code, okay, codes for one amino acid. The same goes for codon and anticodon. Okay, each triplet code codes for one amino acid, meaning to say, we take an example. Okay, CTT. Okay, you know C is one of the nucleotide base. Okay, T, yes, C is senin. Is it senin? Okay, and then T is thymine. So CTT codes leucine only. Okay, so meaning to say each one triplet code will only code for one amino acid. And then each triplet code is degenerate. Okay, meaning to say one amino acid can be coded by more than one triplet code. Example, leucine can be coded by CUU, CUC, CUA, or CUG. I hope you are not confused between the fourth and fifth. Okay. Next, each triplet code is non-overlapping, meaning to say they are always complementary on the other strand. For an example, okay. Oh dear. Pointer option pen. Okay, now if on the DNA, okay, we have TAG. Okay, on the complementary mRNA, it will be AUC. Thymine correspond or complement with adenine. Okay, then adenine on the DNA complement with uracil on the mRNA. Remember, RNA, they don't have adenine. Sorry, they don't have thymine. The thymine is replaced with uracil. And then guanine with what is C? Was it cytosine? I can't recall. Okay. Same goes that you know that DNA, it has two strands, right? Okay, it has two strands. Now let's say this strand here, okay, is A, okay, T. The complement strand here, okay, will be T, okay, T, A. Okay, so this is what it meant with triplet code is non-overlapping. Okay, either on both strands of DNA or between one strand of DNA with RNA. Okay, so if this is the triplet code on a DNA, okay, what would you expect the codon, sequence of codon in the mRNA? Write your answer here. Okay, pause the video. All right, okay, so it should be A with U, T with A, C with G. U, A, sorry, U, A with thymine. Okay, yeah, so U, A, G. This is U, A. Oh, susahnya mau tulis. Okay, this is G. Okay, now codes carried by DNA is called triplet codes. Okay, A, G, C, T, and mRNA carried complementary codes. Okay, or mRNA nucleotide triplets called codons, which is A, G, C, U. Now, class, the term codon is also used for the DNA nucleotide triplets along the non-template strand, like what I mentioned earlier. Okay. These codons are complementary to the template strand, thus identical in sequence to the mRNA, except that they have a T for the DNA, wherever there is a uracil in the mRNA. For this reason, the non-template DNA strand is often called the coding strand. Now, tRNA carried corresponding codes to the mRNA, and the corresponding codes on the tRNA is known as anticodon. Okay, class, anticodon, codon, and triplet codes, they are not in full capital letters. Okay, I purposely put capital letters here to make it highlighted to you. Okay, these are all small letters. Now, start and end of a coding sequence from a cistron is determined by specific codons. Start codon is TAC in DNA and AUG in mRNA. The start codons, they serve as a start signal in the translation of polypeptides. Okay, transcription, translation, I will further explain that to you in our tomorrow lesson. Okay, now the thing is, at this stage, start codon, okay, in DNA is TAC, and this TAC, okay, it codes for the synthesis of an amino acid. That amino acid is known as methionine. You have to memorize this. Okay, and start codon in mRNA is AUG. Next, stop codons. Stop codons are UAA, UAG, UGA in mRNA. Okay, what are stop codons? They are codons that serve as stop signal in the translation of polypeptide. Meaning to say, during translation, okay, when the reading of these stop codons occur, okay, on the ribosome, in the mRNA inside the ribosome, the synthesis of the polypeptide will discontinue. Okay, that's why, as I have said here, the stop codons serve as stop signal in the translation of polypeptide. If translation is keep on bugging your head, what is that? What is that? Hold your horses. We continue that later on. All right. Okay, now, what are the stop codon in DNA? I leave that question to you. And does tRNA has stop anticodons? Stop anticodons? No, it does not. Okay, how? Why? Just keep to yourself that tRNA does not has any stop anticodons. Okay, I will disclose that later on in the translation process. Okay, now this is triplet codes which can be found in DNA. You have this table in your module, except that it is blank. I purposely let it be like that because I want you to construct your own triplet codes table. Okay, so this is the first base here. You know that triplet codes or genetic code consists of 3 bases, right? So the first, second base, and third base. Okay, now it's pretty easy. You start with T, C, A, G. Okay, on the second or on the first, they should be identical. TCAG, TCAG, and then here also TCAG. You don't do like here you write TCAG, and then over here on the second base you write GC, GCAT, like that. No, it has to be identical. The sequence must be identical. So TCAG now. So T, if this is T, this is T, and this is T, you have T, T, T. Then T, T, C. Why? The T from here, but let me change the pointer in color, green. Okay, next, T, T, C. So you have T, T, C. Again, then T, T, here, A. Then you have T, T, A over here. Next is T, T, G. How do you get T, T, G? T, T, T over here, T over here, and the G over here. And you get T, T, G over here. right? So how do you get TCT over here? Here. Okay, again, T from here, and then C from here, and T from here. So T, C, T, right? So you do this one by one. Okay, it's pretty easy, right? To construct your own DNA codons or triplet codes. Okay, now, as what I've mentioned earlier, each triplet codes only codes for one type of amino acid. So TTT, okay, TTT only codes synthesis of phenylalanine. TTC only codes synthesis of phenylalanine. And I mentioned earlier that amino acids are degenerate. The triplet codes are degenerate, meaning to say one type of amino acid can be synthesized or coded by more than one type of triplet codes, right? Like phenylalanine, you can get phenylalanine from TTC or TTC, TT or TTC. All right. Okay, so you complete your table. Okay. Now, I want to highlight here ATG, which is the methionine. TAA, which is the stop triplet code. TAG, stop triplet code, as well as TGA, stop. So this one you have to memorize: ATG, TAA, TAG, and TGA. All right. Now, next is LEU. I already write down the keyword here. Okay, LEU is leucine, capital I, okay, small l, small e, isoleucine. Okay, the methionine, valine, serine, right? These are all the keyword for this particular short forms of types of amino acids, right? So complete your table for the DNA codons or triplet codes. The same goes for the mRNA codons. mRNA, you know already. Okay, thymine is replaced with uracil. So UC, AG, okay, and then you see AG here, and you see AG over here. Okay, so U, get U here. Next, C. Okay, U from the second base and first base, and third base is C over here. So you get C, right? So apparently, if you notice, okay, the sequence of the amino acid here between the DNA codons or triplet codes with the mRNA codons, they are the same. You have phenylalanine here with leucine. Okay, the same goes here. Phenylalanine with leucine. You notice that? It's the same. Only thing that change is the base here from thymine to uracil. Itu yang beza dia. Lain-lain sini semua sama. All the same. Semua sama. Okay? Now, since uracil is already replaced, thymine is replaced with uracil, so the start codon in mRNA will be AUG, which code for methionine. Sama seperti DNA tadi, except the the genetic code is different. It's AUG for mRNA. Okay, stop codons will be UAA, UAG, and UGA. Right? Now, this non-polar, polar, okay, positively charged, negatively charged, this you have learned in the first sem already. Characteristics of these amino acids. Okay, so do you need to know these characteristics in the third semester? No, you don't have to. Okay, you need to memorize all these. If you can memorize, that's better. If you can't, it's okay. But what you need to know, you need to memorize is these codons here that codes for starts, as well as the synthesis of methionine then stop codons 2. All right. Okay, now genetic code in DNA known as triplet codes. mRNA is codon and tRNA is known as anti-codon. Okay. Let me change the color of this pointer. I think better use the highlighter. Now, okay, now the DNA, let's say we have this sequence of triplet codes, okay, on a DNA, and it's being read from the 3rd prime to the 5th prime. So the codons on the mRNA will be complement to the triplet codes on the DNA. So it will be read from the 5th prime to the 3rd prime direction, and it has to be complement. So adenine with thymine, you can see here. Then G with C, C with G. Okay, and then next to the anticodon on the tRNA, it will be the opposite. So from 3rd prime to 5th prime, which is very similar to the DNA. Except that, again, don't confuse, we don't have thymine in RNA. tRNA is an RNA. Okay, so UU, GC, and so on. Right, now codon in the mRNA need to be written in the format 5th prime to 3rd prime direction, like this. Okay, codon on the mRNA, always like this, 5th prime to 3rd prime. Now transcription is a process where the template on DNA strand, the leading strand, is being read. Okay, and then the direction is always from the 3rd prime to the 5th prime. Formation of mRNA, okay, or the direction of transcription. Transcription is a process where we have mRNA being synthesized. Okay, the direction is from 5th prime to the 3rd prime. Then in translation, okay, the reading of codon in mRNA is from the 5th prime to 3rd prime direction, and then the corresponds anticodon on tRNA is 3rd prime to 5th prime. Okay, like what I mentioned here as well. Okay, now this is 2013 repeat paper. What do you think the answer is? Yeah, I'm having sore throat already. Okay, so you figured it out. Pause this video first. All right, done. You get your answer already? Okay, let's check your answer. Okay, the answer is B. All right. Next, 2014. What do you think the answer is? Pause the video. Done. All right, this is the answer. Okay, 2014. Okay, the sequence of blah 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 blah, blah corresponding anticodon and mRNA respectively. Okay, just now I already mentioned, right, the reading of the codons and such, this and such, the directions. So what do you think the answer is? Pause the video first. Alright, done. You get your answer already? Okay, let's check. It's B. Alright. Okay, repeat paper 2015. What do you think the answer is? Okay, done. Alright, this is the answer. Okay. 2017. What's the answer? Done. You get your answer already? Alright, so this is the answer. Right. So that's it for today. Okay, tomorrow we continue with transcription and translation. So happy studying. Bye.